Just for a brief introduction, Dr. Decker is an associate professor of pathology and immunology at the Baylor College of Medicine, and we really appreciate your thoughts as a leading expert in the field. So the first question is, how does the human immune system work?
There are two big arms to the immune system. There's the innate arm that no one ever talks about or even knows exists, and then there's the adaptive arm, which is CD8 and CD4 cells and B cells. We won't get too much into the innate immune system, but it doesn't involve any memory. So all these other mechanisms that you know about as part of the adaptive immune system, those are for problematic pathogens that get past all your innate defenses, and then they need to be addressed specifically by the adaptive immune system. So something like 95% of pathogens, bacteria, viruses, all that stuff, we don't call them pathogens because they can't get past innate immunity, so they don't even show up. We don't even know when we're infected by them. They don't make us sick. They just get taken care of by the innate immune system. And you don't even develop antibodies or T cells against them because it's not necessary. So let's get that out of the way. So then we're into the 5% of pathogens, or specifically viruses, that are pathogens because they do get past innate immunity and they do require our adaptive immune system to clear them. So then there are two main mechanisms by which the adaptive immune system will get rid of viruses. One is through T cells, and two is through antibody responses. So the way an antibody response works is that little parts of the virus are actually recognized by the immune system, and you end up making what are called antibodies against them. And antibodies are molecules that bind really tightly to the pathogen, in this case a virus. And so one end is bound to the virus, the other end is recognized by the immune system. And that allows viruses to be cleared, it allows them to be neutralized. If antibodies bind to the important parts of the virus that allow the virus to attack cells. So basically any virus recognized by antibodies, once an antibody response exists, will become coated with antibodies and can't infect anything. It'll just get gobbled up by the immune system. So that's antibody-mediated immunity. T-cell-mediated immunity is a little bit different. That involves attacking and killing your own cells of your own body that are making virus. So antibodies exist outside of cells. They circulate in the blood and in tissues, but they don't go inside cells. And so if you really want to clear a virus and create sterilizing immunity, you need what is called a T-cell response that actually recognizes cells that are infected with the virus, and the response, just like an antibody, is very specific to parts of the virus. And you have every cell in your body with a nucleus has a surveillance system that allows all the proteins, including pathogen proteins, inside the cell to be surveilled by the immune system. So that's what T cells do. They're constantly surveilling cells, looking for little parts of foreign molecules that they can recognize, and in this case, virus-specific foreign molecules. And so when those are seen by a T cell will actually kill your own cell that's making the virus. And as you can imagine, T-cell mediated immunity you want that to be well regulated and well controlled, because if it's not, you can imagine you could get very sick if you start killing off all these cells of your own body in a non-specific way. So it's a tightly regulated process, but required to generate sterilizing immunity once you're infected with a virus.
For a second question, it is, what is the difference between the terms coronavirus, SARS-CoV-2, and COVID-19? Or in other words, what is the pathogen of the COVID-19 disease?
That's a great question. So SARS-CoV-2 is the official name of the pathogen. That's the name of the virus. COVID-19 is the name of the disease. And coronavirus, there's a whole class of viruses called coronaviruses, and SARS-CoV-2 is a coronavirus, but there are also a lot of other coronaviruses. So the word coronavirus technically is what we use to describe the whole class, but since this SARS-CoV-2 is the only coronavirus everyone is talking about or interested in, it's sort of developed as a synonym for COVID-19. So if you say COVID-19, people know you're talking about the disease that is currently raging around the world. And if you say coronavirus, most people just take it that you're talking about SARS-CoV-2 because you typically are, right? So those are the difference between those terms, and they're sort of largely interchangeable.
So just for us to get some background, what is a brief history of the coronavirus and what severe diseases are actually caused by the virus?
Okay. So there are, including SARS-CoV-2, there's 7 different coronaviruses that infect humans that we know of. The coronavirus in general, for most of human history, again, what we know, or what we think we know, was basically a cold virus. So there are 4 circulating coronaviruses that cause colds. And for whatever reason, you don't develop, in general, long-lasting immunity to these coronavirus infections. I don't think the reason for that is really well understood. Or even if what I just said is true, isn't really well understood. The dogma has been you keep getting colds over and over and over because of these 4 different circulating viruses, to which immunity will wear off after, I don't know, not a very long time, maybe a year or 2. Again, not entirely sure if that's true, but that's what's largely thought. Relatively recently, these very severe forms of coronavirus disease have emerged that have impacted human populations in a really bad way. There are hundreds, probably thousands of coronaviruses circulating in other species. So not just, they're the ones that directly infect humans, but then there are these other hundreds or thousands, or we don't really even know how many, that are circulating in lots of other species. And every once in a while, a new virus will make a jump from animals to humans. There'll be a new mutation, or an animal will be infected with multiple coronaviruses, and they'll like recombine in a way that allows them to infect people. And that happens every so often. And when something like that happens, you get the outbreak of a new disease. And in the case of these 3 coronavirus diseases that are very serious, they've caused pretty bad disease in people. So the first one that emerged was called SARS. We had sort of a mini pandemic, and everyone was sort of panicking just like they are now, except that was much more easily controlled. That virus did not spread as easily as this one. And there didn't seem to be the phenomenon of lots and lots of asymptomatic people. So, if you had it, you were sick. So you were easy to find and trace and quarantine. So we were able to stamp that out without a global pandemic. That killed around between 3,000 and 4,000 people total in China and also in Canada. But we were able to largely control that. So then a few years later, an even less easily transmittable coronavirus disease arose in the Middle East, and that was called MERS. Middle East Respiratory Syndrome was the name of the disease and the virus, and that was linked to camels. And again, it really didn't transmit very well from person to person, and that ended up, and it was largely restricted to the Middle East, where could be direct camel to human transmission, and that killed about 900 people. Those are the 2 bad coronavirus diseases that we had as predecessors, but because they weren't able to spread, they weren't really well adapted to people, and we were able to stamp them out before they could get really well adapted. And then this came along, and this has been really devastating, right? It's for whatever reason, luck. It's probably luck, because we're probably being bombarded with new viruses all the time, hundreds, thousands, millions even, and most of them end up being harmless. So eventually you get just that right combination of different mutations and different recombinations that end up making a virus that is very pathogenic and not only spreads widely like this one, but in many cases can also cause severe disease. So that's maybe not so brief, but that's a history of coronavirus infection.
Now to get into the vaccines, how do the contents of different vaccines differ so that they're specific to their respective diseases?
That's a great question. So are you talking about vaccines in general or coronavirus vaccines? Vaccines in general. Right. So for a vaccine, to train the immune system to recognize the pathogen, the vaccine actually has to share proteins in common with the pathogen. So the best vaccines are what we call attenuated vaccines. That's the actual live pathogen. And so when you get it, you set up a real infection, but attenuated in such a way that it's missing important virulence factors that allow it to make you sick. So, there's a live anthrax vaccine and that can't turn around and kill you because it's missing important virulence factors. One of the polio vaccines is an attenuated polio virus. It has some mutations in it so that it can set up an infection and the immune system can respond to it, but that it doesn't make you very sick. So the best vaccines actually share almost everything in common with the actual pathogen, except for a couple of virulence factors that really cause disease. A lot of vaccines are what we call they're the actual pathogen, but they're killed. They're either heat killed or they're formalin fixed. And so those don't set up a real infection. But when you inject them into a person, you can develop an antibody response to all of the proteins on the outside. And the flu vaccine, the seasonal flu vaccine that we get every year is exactly that. It's a formalin fixed flu virus that you generate an antibody response against. So those kinds of vaccines, either live attenuated or killed vaccines, are the actual pathogen itself. So it has all the proteins it needs. It's not even a mimic of the actual pathogen. It is the pathogen. So the immune response is actually responding to all of the real proteins of that pathogen. So then there are other kinds of vaccines called subunit vaccines, where we're not using the whole pathogen, but we're using parts of it. So subunit vaccines are just specific parts of the pathogen, like one molecule, against which if you make an antibody response, you can get protective immunity. But you always have some actual part of the pathogen. Now, these RNA vaccines that are brand new and were created for coronavirus actually work differently. So they're very revolutionary. They don't work by actually putting part of the pathogen into you. The mRNA vaccine is actually just information. It's injected into you with a special lipophilic coating that allows it to get into your own cells. And once it's in your own cells, the RNA provides information to that cell on how to make a critical coronavirus protein. So you're not supplying the protein, you're just actually supplying instructions so that your own body will make that coronavirus protein. And it's the critical coronavirus protein against which you need an antibody response to stop the coronavirus from infecting you, and that's called the spike protein. So a vaccine always has to have either parts of the actual pathogen, or at least it has to have information that allows those parts to be produced inside your body.
Our next question would be, the FDA recently approved the Pfizer and Moderna vaccines for emergency use. So how do these vaccines work?
Yes. So one thing that is super interesting about these vaccines is they're basically identical. They're basically the same vaccine. They are coated with a lipophilic particle. So what's probably mainly different about them is Pfizer has its own lipophilic particle delivery system, and Moderna probably has its own proprietary lipophilic particle delivery system. That's probably the main difference between the two. And it's not even a significant difference. All we care about as vaccine recipients is that it works, right? That it gets into your cells, provides the information to the cellular translational machinery to make spike proteins so that you can make an immune response against the spike proteins. But they both work exactly the same. There may be small differences in the mRNA. So the information encoded by each of those vaccines may be slightly different. For all practical purposes, they're essentially the same. They both make spike proteins inside your cells that then get out and stimulate B cell and T cell responses.
What ways are the COVID-19 vaccines in any way different or similar from traditional vaccines?
Yes. So I think they're enormously different. I mean, they're similar in that you know, the immune system is the immune system is the immune system. When you present foreign proteins to the immune system, it makes protective responses. But up until now, these are the first vaccines where you are not really generating any physical part of the pathogen at all, just information as to how the important immunogens should be made in you. That's really revolutionary. And I think you can appreciate very different from what we've had before.
You kind of just touched on this, but how have the different COVID vaccines been developed, and in what ways do they differ from previous vaccine development?
Right. The technology of providing information to your own cells isn't a new idea. I mean, that was thought up a really long time ago. And the first thing that was thought of was using not RNA, but DNA. So the problem with using DNA, I mean, it's more of a theoretical problem than like a real problem. But the theoretical problem with using DNA is that it's actually very stable. And, you know, it might not just sit there and make the proteins you want. It might break and get into the nucleus of cells and integrate and end up causing cancer. For that reason, DNA never really took off. So the logical thing was RNA, because that can't integrate into the genome, but RNA was really unstable. So what's happened over the last 20, 30 years is that people have worked on stabilizing the RNA and coming up with good ways to get it into cells once it's injected. But since we had the big crisis and the technology had been in development for 20, 30 years, it got pushed through very rapidly by our regulatory authorities.
For our next question, what is herd immunity and how will the implementation of vaccines help us achieve it?
Right. So if I am sick with coronavirus disease and I'm walking around coughing on everybody, and nobody's immune, I will easily spread the disease, right? I'll spread it to you, you'll spread it to your friend will spread it to his friend, and his friend may then spread it to, you know, his other friend's grandmother in a nursing home, right? There's nothing to stop it. Now, if all of those individuals between me and the grandmother in the nursing home are immune, I cough on you, you're immune, so you don't end up coughing on someone else who doesn't cough on someone else, who doesn't eventually end up coughing on the person who isn't who can't be vaccinated, who doesn't have a good immune system, that can't really respond to a vaccine, or for whatever reason, you know, has a lot of health problems and isn't appropriate to vaccinate. So herd immunity is providing a protective immune response in enough people so that the people who can't be vaccinated and can't be protected are protected, because they never can come in contact with someone who's sick, because, you know, statistically most of the people you come in contact to are vaccinated. So the number of people that need to be vaccinated to achieve herd immunity is different from pathogen to pathogen to pathogen. It sort of depends how infectious the pathogen is. So the most infectious pathogen there is the measles virus. It spreads incredibly easily. It's really super efficient. So to achieve herd immunity against the measles virus, you have to have more than 95% of the population vaccinated. But then you have less efficient pathogens that don't spread so easily. And so for those pathogens, you can have lower numbers of people vaccinated to broadly achieve herd immunity. People have estimated that you only really need to get 75% of the population vaccinated to get herd immunity against the coronavirus. I think that sounds a little low. You know, yeah, I've seen the math, but that sounds a little low to me. I think in general, what I tell my students is that you start to get herd immunity for whatever, just generically for most diseases, around about 85% of the population is vaccinated. When you get to that number or beyond it, it becomes statistically improbable for a virus to continue to replicate itself when 85% of the targets it bumps into are immune and can't continue to pass it along. So that's sort of a good rule of thumb, about 85%.
So for our next question, how long will an individual be protected after vaccination?
Right. So the only real answer anyone can give you to this is, I don't know. Because, we vaccinate someone and then we sit back and we watch. How long are you protected? 5 years, 10 years, 20 years? You don't have to wait for someone who's vaccinated to actually get sick just until correlates of the immune response disappear. The only answer someone can really give you is, I don't know. But I can give you my opinion. And my opinion is that these vaccines are going to generate immunity for quite a long time. So the worst vaccines we have that are generally used are the flu vaccine, which gives you protective immunity for about a year. So I'm very comfortable saying at least a year. But that said, this is a much better, more robust vaccine. It provides, so by virtue of the fact that it's RNA, so RNA itself is very immunostimulatory. So you have parts of your innate immune system can actually detect RNA, and you're sticking a bunch of RNA into you, and you get production of an immune mediator called interferon. And interferon is one of the things that makes your arm hurt. But also it turns on different parts of the immune response in a way that isn't done by vaccines that don't work as well. And it allows you to get a longer lasting, more substantial response. So like I said, we don't really know, but it wouldn't surprise me if we got many years of protection from this vaccine. Now, will we get 30 or 40 or 50 years like we would get from like from really getting a disease against which you make a permanent, durable immune response? Probably not. So, I mean, you're not going to, I wouldn't imagine you'd end up getting like 40 years of immunity, but I can easily envision getting 5 to 10 is my best guess.
Why do certain vaccines require more than one dose?
So the real answer is that when you're developing a vaccine, you decide this threshold of response is correlated with immunity. So if you get antibodies at this certain level against this certain protein, we're calling that immune. That's what we see in our studies. That's what we see in our animal models. That's what we see by looking at people. So then you go and you start developing a vaccine and see how much vaccine you have to give and how many times you have to give it to get to that level of immune response that you've predetermined is your correlative immunity. And that is really completely empiric. You give one shot, you get there 95% of the time, you say, great, we're going with one shot. You give one shot, you only get there 40% of the time, you say, not enough, give another one. So the real answer is it's sort of determined empirically. The immunological answer is that the different compositions of any vaccine will give you certain signals that turn on the immune system. And the more signals you get, the better immune response you get, the fewer different vaccinations you have to give. So something like a live attenuated infection that sets up a live infection, replicating, turning on all different kinds of parts of the immune system, pretty much you just need one vaccination. And then subunit vaccines that have fewer signals that don't replicate, those are the ones where you typically have to give more doses. So it really depends on the number and kind of signals the immune system gets from the vaccine is the immunological answer, but the real answer is whatever works. So for the coronavirus vaccine that Pfizer made, they determined to get super sky-high titers of antibodies, you need 2 shots. That was sort of predetermined. But in their clinical trial, they figured out that first shot protects you practically. So you still need the 2 shots to get that absolute sky-high. And the titers you get from 2 shots of the coronavirus vaccine are higher than those of convalescent patients that have actually had the disease. I mean, they really wanted super sky-high titers, and that's where they set their bar. So even the sort of variable titers that you get after 1 shot, some people respond at this level, some at this level, some at this level. It's good enough for most people in most cases. And then they come in with that second shot, that booster, and everyone just shoots up to these ridiculous titers, and almost certainly after a week or two after your second shot, everyone has responded really well, and the protection probably goes up from 95% to something very close to 100%.
So our next question is there a possibility that the currently approved vaccines will prove ineffective to the new strains of virus, such as the one in the UK?
It doesn't seem likely that it will be ineffective. So what we've seen from the coronavirus is that it mutates the way normal things mutate. It's not a hypermutating virus. It has basically, it's very faithfully replicates itself, and doesn't use mutation to its advantage. So this new, more infectious strain that's come out of the UK, it's due to one amino acid change that the best guess is that makes it bind tighter to its receptor. So that makes the spike protein bind tighter to the cellular receptor, which is called ACE2. And that allows it to be more infectious. But so the question is, does that one amino acid change, does that negate immunity? And the answer is almost certainly not, because it's a big protein. It's got hundreds of amino acids, hundreds of little parts against which you can still make antibodies. But here's the really cool thing about mRNA vaccines. Let's say it was discovered that this one amino acid change was really important. You don't have to start over, like make a new protein, take it through clinical trials and all. You just make that change in your RNA, and then you release a new vaccine that has both RNAs in it, make against what we have and what the virus came up with. And so even if it does end up being that's the case, that this one amino acid change abrogates all meaningful immunity, which again seems super unlikely, with this new technology that can be addressed pretty quickly. So all seems far-fetched and unlikely that this one amino acid, or that and in general, the low level of mutation that this virus has is going to be enough to defeat vaccines.
I know you talked about your arm hurting, but what are some of the possible dangers for recipients of the vaccine, such as the current COVID vaccines, and what's been done to mitigate them?
Vaccines have to be very safe. Healthy people take vaccines, and typically in society, your risk of getting whatever disease you're being vaccinated against is very low. So people won't put up with any safety issues. So all the vaccines on the market today are very, very safe. There's concern that the coronavirus vaccine might not be as safe as any other vaccine because it was rushed. And so the opposite is actually true, in that the clinical trials carried out for the coronavirus vaccine not only used young, healthy adults, which is what other vaccine trials used, but they also used elderly people, because it's very important to protect against elderly. And so in general, those trials showed very good safety. Now, the vaccine is a little harsher than other vaccines. I mean, so there are some side effects of it that are maybe a little more inflammatory than you would normally get in a vaccine, because it's this new kind of RNA vaccine that we were talking about. So it's gonna hurt a little bit, but no more than a tetanus shot, which of course hurts a lot, right? And the second shot you get one, and your arm's gonna hurt the next day, kind of like a tetanus shot. You might feel tired. The second one may actually make you a little bit sick, as you have an immune response to your original, to the original response that you set up. You're getting a booster, so you might actually feel sick, like you might have a fever, you might have aches. It'll be like having a real infection, except you are not having a real infection. You're just having the immune response. So that's all not wonderful, but it's not long-term dangerous. So you end up getting this really great immune response without the possibility of having some kind of long-term problem. Now, there are some people in general, and these people know this about themselves, that don't tolerate vaccines very well. Any kind of immune response, they're kind of like kind of auto-inflammatory people. They have, autoimmune conditions, or they just don't handle inflammation well. Those people usually know that about themselves. And so if you're one of those people and you've had bad reactions to the flu vaccine or other vaccines in your life, you may also have an unusual inflammatory response against this vaccine. But in comparison to any other kind of vaccine, it is just as safe, if not safer. And the studies that we've done so far all bear that out. In the 75,000 people who were in the clinical trial, they all did really well, only had short-term issues. And in the post-marketing studies that we've had so far, there really have been very few reported complications. I think in December, we vaccinated about 2.5 million people, and 2 anaphylactic events were reported. And neither one of those ended up really being very serious or life-threatening. So as far as we know, this is a very safe vaccine.
How do vaccines affect different people? Or would it have the same success rate for people of different ages, sex, immunological status, or so on?
Right. So that's another really great question. Also a little bit complicated because you have different variables here, and you're also asking about vaccines in general, plus the coronavirus vaccine, I guess, specifically. So I'll start with sex. So there's a theoretical reason to think that women would respond better to a vaccine than men. I say it's just theoretical because when you develop a vaccine, you make it so that it is good enough for everybody. So you don't scale it down so that it has a differential impact on men and women. You scale it up so that it works in everybody. So that's a theoretical. So women's immune systems work better than men's without going into the biology behind it. It's a fact, just accept it. Practically, I don't think I've ever seen a study that has suggested that men and women are responding differentially to vaccines. So that's more of a theoretical thing. Now, ages and immunological status, that makes a huge difference. So if you have severely compromised immunological status, your ability to make adaptive immune response is also compromised. So cancer patients who are getting really high doses of chemotherapy, people with really severe autoimmune disease that are taking super high dose steroids, those kinds of people may not respond at all. So those are the kinds of people we want to protect by building up herd immunity. There's no problem vaccinating them. They just may not form a very good response or any response at all, or if they do, it may not last very long because they're severely immunologically impacted. And the corollary to that is also age. So as you get older, your immune response gets less and less and less and less efficient. Like I said, the coronavirus vaccine is different. It gives you a lot of signals through the RNA itself, this big giant bolus of RNA you're sticking in, that generates interferon. And what was seen in the clinical trials, and the clinical trials were carried out in individuals up to 85 years old, it works practically as well in elderly people as it does in younger people. There may be a small drop off, but not much. So even if you're old, if you supply a lot of inflammatory signals, you can still make immune responses. So in general, vaccines that have fewer and weaker signals will not work as well in older people, but vaccines that still have very robust signaling will continue to work well.
And for the next question, what advances in medical technology have led to the expedited rate of vaccine production and increased clinical trials? And is it possible that the rush to produce a vaccine could have compromised safety?
Right. So what made vaccine production here, what enabled this to go quickly, was that we're in the middle of a global emergency. Our government did stand down the regulators and say, let's get this through as reasonable as we can, so long as the companies that are developing the vaccines are doing everything that they can do. There's a really concerted effort to move as fast as possible. That's why this went fast. And I'm grateful that it did. So is it possible that the rush to produce a vaccine could have compromised safety? There are 2 reasons why I'm going to say no. So one is that in general, I still trust our regulatory bodies, they're still staffed with good people who actually care about public health and they want to do the right thing. And in general, the companies are also staffed with good people who want to do the right thing. If there was a real issue with this vaccine, it's going to come out. The whole world is going to get this vaccine, and fast. You know, billions of people are going to get this vaccine. If they did something wrong, it's going to get caught fast. So that's also what's keeping them in line.
In your opinion, how important is getting a COVID-19 vaccine and why?
Right. Have you enjoyed the last year? Been a good year for you? No, it's been a terrible year. And the only way this ends and everything goes back to normal is when cases not start to drop, but drop. They go to close to nothing and we stay there. The only way that happens, the only way we all get our lives back is that we all get vaccinated. Young, healthy people like you, you're just not getting sick. The numbers are tiny. But we have a moral obligation to protect the elderly in our society. So.